Dr. Fong, you were part of the team that brought the first immunotherapy drugs from the labs to clinic. It would be great if you could, talk a little bit about this fascination with the immune system and your journey in being able to treat cancer with your own immune system.
Well, my interest in focusing on cancer is a bit personal. My father died of cancer while I was in college, so I decided to go into medical school hoping to work on new treatments for this disease. And while I was in medical school, I actually became intrigued by the complexity and power of the immune system while taking the introductory immunology course during my first year in medical school. If the immune system could target certain types of infections with fine specificity, why couldn't we use it to treat cancer? And, at that time I was in med school, there were some immunotherapies just starting to make their way into the clinic, these cytokine therapies and cancer vaccines. And so, I decided for my research training in fellowship to really dive into this. And so I decided to work on immunotherapy for solid cancers, including prostate cancer. I recall one of my professors at the time telling me that, this approach would never work. Nevertheless, I worked with groups at Stanford, including Dr. Ed Engelman, on a dendritic cell vaccine for prostate cancer, and this ultimately became an FDA-approved therapy called Sipuleucel-T or Provenge. When I moved to UCSF, we continued with our interest in immunotherapy, and I had a chance to work on the first in-human trial with another immunotherapy. This was an anti-CTLA-4 antibody, which is now called ipilimumab and is also FDA approved. And, since that time, there have been other therapies really making their way into clinic, especially these anti-PD-1 antibodies that we've worked on, and it's been a very wild ride ever since.
So it has probably taken decades of basic research to understand the fundamentals, of our immune systems, and then probably overlay on them the fundamentals of cancer, and then further overlay how the two of these things work together, and then try to come up with a solution. It's truly fascinating. Do you think this is the penicillin moment for cancer?
Wow. Yes and no. Yes, in that, our approach to treating cancer has forever changed, not unlike when penicillin came out. Just as patients with bacterial infections now receive antibiotics, I think the majority of cancer patients in the future will receive immunotherapy. In fact, we now teach our medical students about cancer immunotherapy in their introductory class as they actually start out in medical school. So it's really changed our way of thinking. Unfortunately, the challenge is that our current immunotherapies do not work as well as penicillin. Only 10 to 30% of our cancer patients benefit with our current immunotherapies when they work. For those patients, they may have very durable responses, including some folks being cured. Our challenge is now to get immunotherapies to work in more people.
Dr. Fong, I'm going to ask quite a broader question here. So could you please talk about some of the most effective cancer immunotherapies that we know today?
Well, there are three areas where immunotherapy has had great success at this point, and those are immune checkpoint inhibition, bispecific antibodies, and the third are CAR T-cell therapies. With immune checkpoint inhibitors, these are the antibodies that target CTLA-4 or PD-1 or its receptor PD-L1. These different molecules are essentially brakes on the immune system, and by giving these antibodies, we're blocking those brakes so that we can turn or revive the immune system to target the cancer. These antibodies can lead to responses in a broad range of cancer, and the list of approvals where we can administer these drugs is getting longer by the day, it seems. We've also found that combining these checkpoint inhibitors, such as anti-PD-1 and anti-CTLA-4, we can actually see better response rates in certain diseases, such as melanoma and kidney cancer, and for some lung cancers. With regards to that second area, bispecific antibodies, these are drugs where one end of the drug targets the cancer cell and the other end targets the immune system. By pulling these immune cells into the tumors, we're banking on these immune cells killing the tumors. And in fact, we've got a drug already FDA approved for B-cell malignancies that does this. The third area are these CAR T cells, or what are called chimeric antigen receptor T cells. With this treatment, we take immune cells out of a patient. We actually engineer those cells so that they have a receptor that targets the cancer cell, and then we give those cells back to the patient. So these immune cells are now hardwired to recognize the cancer. And we now have multiple CAR T cells approved that are targeting B-cell leukemias and lymphomas.
Dr. Fong, since you're mentioning, you've already just mentioned CAR T cells, I'm going to take an audience question on this. The person who's listening wants to know, can T cell therapies be used against common solid tumors?
Well, right now that's a very active area of investigation. As I mentioned, the CAR T cells that we currently have approved target B-cell malignancies. So these are leukemias and lymphomas. With regards to solid tumors, that's proved to be a bigger challenge. And one of the challenges is actually finding a good target on a solid tumor, and there are multiple groups that are working on this, and groups are also looking at ways where we don't rely on just one protein, but we might rely on integrating recognition of multiple proteins to target and kill a tumor cell. The other element is, with regards to solid tumors, the tumors themselves are very inhospitable for immune cells, and so we're also thinking about ways to make these CAR T cells resistant to those suppressive mechanisms so that they actually work better there. And so this would be an area that I'd really recommend staying tuned as these new cell therapies come into the clinic.
Absolutely. So we have another question on the same lines on CAR T and engineering of the genes for these re-engineered T cells. So the person wants to know, can Dr. Fong talk about CRISPR and how it can help cancer immunotherapy?
Yeah, so CRISPR is a new way to edit genes in cells. And, CRISPR therapies are being developed to target specific cell types within the body. CRISPR has also been used to engineer cells outside the body. In this context, taking out, for instance, T cells and editing in a receptor that's targeting the tumors. And so this is a new technology that's just made its way into the clinic. And, over time, I think as we learn more how to use CRISPR, we'll more than likely see this being used in more and more clinical trials. And it should make engineering of the cells easier than the methods that we use today.
Thank you, Dr. Fong. Going back to our initial trail of questions, we were talking about checkpoint inhibitors and CAR T cells. So, as you said, Doctor, immunotherapy does not work for all tumors. So it follows that other drugs can combine with immunotherapies. So it would be great if you could talk a little bit about the impact of immunotherapies on the scope of combination therapies.
Yeah, so I mentioned briefly that we're now combining anti-CTLA-4 and anti-PD-1 for some cancers. Really an area of active research is identifying other immunotherapies that we can combine together, including combining with these existing antibodies that we have. But importantly, we've also found that we can combine immunotherapies with our conventional cancer treatments, such as chemotherapies and targeted therapies. As it turns out, these cancer therapies that we've used for years can also affect the immune system. And so now we actually use a combination of an anti-PD-1 antibody with chemotherapy for the first-line treatment in lung cancer, and we're also now able to use that in some breast cancers. With regards to targeted therapies, we have these drugs that target blood vessels, what we call tyrosine kinase inhibitors, or TKIs. What we found is you can actually use these drugs to combine with anti-PD-1 antibodies, and this is now an FDA-approved frontline therapy for kidney cancer. And so as we learn how other cancer treatments modify the immune system, we're looking forward to more and more combinations with immunotherapies in the future.
Thank you, Dr. Fong. So what do you see? I know you're talking about combination therapies and targeted therapy. So what do you see as the next major milestones in cancer immunotherapy, say, over the next 5 years? And we'd also like to know about the programs at UCSF Helen Diller Center that patients should keep track of.
Yeah, so over the next 5 years, first, I hope that we'll see new drugs targeting other immune pathways gaining traction. Right now, a lot of interest is around the immune checkpoints, but there clearly are other pathways that are also incredibly important. And, as we get more of those drugs available, this will add to our treatment toolkit going forward. Second, I think our understanding and our ability to identify who can respond to these immunotherapies will improve over the next 5 years. And so, this is important for us to not only give our existing immunotherapies to patients who we believe to respond, but it would also be important for us to be able to select and identify patients who might need combination therapies or other treatments added to their immunotherapy. Ultimately, we'd love to be able to tailor immunotherapy to each patient. This idea of precision cancer treatment, and I think that would be possible in immunotherapy as well. Finally, I think over that time frame, we'll see CAR T cells and bispecific antibodies that will work on more cancers. Right now, we only have those treatments approved for B-cell malignancies, as we talked about. But I think, over the next 5 years, we'll see that broaden out to other diseases. In terms of your question about the cancer immunotherapy program at UCSF, this is a program that is focused on bringing new immunotherapies into the clinic. Historically, cancer centers and programs had focused programs on where the cancer arises from. So, for instance, from the lung or from the breast or from the prostate. Our program actually is focused on the treatment modality, which is using the immune system to treat cancer, because there are specific considerations in how we can administer these safely, as well as how we can get them to work better. And so within this program, we have clinical trials that span different diseases. This includes new checkpoint inhibitors, vaccines, bispecifics, and cell therapies. Our mission is to learn as much as we can from each person that we care for. And so to realize that vision, we have clinical and laboratory researchers integrated into this immunotherapy program. What that allows us to do is to perform cutting-edge research on samples from our patients, and really to tease out what these immunotherapies are doing in people with cancer. I'm often asked by people I'm caring for how their immune system is responding to the treatment. We hope with further research that we'll be able to answer these questions in the future, and that these types of discoveries will help guide new immunotherapies and new immunotherapy combinations. And so, it really is a program that has multiple clinical trials going on now, and we have over 20 clinical trials, but we also have a pipeline of even more trials that we'll be activating. But importantly, we hope to not only be administering these treatments, but again, really advancing the field with new discoveries.
Oh, that sounds really interesting, Dr. Fong. I'm going to actually stop my questions now and let the panel take over. Before that, I want to just take one question from the audience. The person wants to know, we have been reading about cancer vaccines. In fact, Stanford has a human trial on for vaccines. Dr. Fong, where do we stand with respect to vaccines for cancer?
So right now, the only FDA-approved cancer vaccine for treating patients with cancer is Sipuleucel-T or Provenge, and that's FDA-approved for men with advanced metastatic prostate cancer. There are studies now focused on a whole host of different vaccines. That vaccine that I described targets a protein that's expressed on prostate cancer, and so it can be administered to a broad range of prostate cancer patients. But one area of research now that's quite active, and we also have multiple clinical trials studying this at UCSF, is to use customized vaccines. And so in this approach, what we do is using a biopsy from a patient, and that could either be one that was done in the past or one that we do for the clinical trial. We actually sequence that individual's cancer to understand what types of mutations are present within that cancer. And because these mutations only arise in the cancer, we think that these would actually make great targets for a vaccine. And so with this sequence information, the companies that we're working with on these vaccines basically create a specific vaccine for that specific individual, so it's customized. And so that vaccine is given to an individual, and it's usually given in combination with a checkpoint inhibitor. And so right now we have a couple of those clinical trials going on at UCSF. I think this is an area of very active investigation, and I would also recommend staying tuned as we get more and more experience with this approach.